Very good afternoon to everyone who has attending the conference. Today, my talk is on paradigm shift in harnessing the importance of biodiversity for human global health using genomics. I'm Subha, and I come from University of Malaya. My lab is animal genetics and genome evolutionary biology. Now, let me take you to the work that we are doing from our lab, where we use genomics technologies to facilitate the conservation and management of natural resources. Now, we are looking at three different components, one from the perspective of host, then another one is a perspective of pathogen, and the third component is environment. It's very alike like in quantitative genetics, you have the phenotype equal to genotype plus environment. So the phenotype here is the host, and we started off in our earlier research many years ago using mitochondrial genome and of course the genome of the organism, genome information. And then we started looking at functional genes and we started asking questions, is there a signature of selection? Is there a signature of adaptation? Whether the genomes are duplicating? And we wanted to know whether there's any functional adaptation based on different geographical location. And as the second component is the pathogen, where we wanted to look at the things that is being affected, the host is being affected from viral, bacteria, and parasites. And we also looked at it from a micro environment, from the pathogen perspective, looking at the pathogen genome and its adaptation and how it survives in the host. And this is where we started looking at host pathogen interaction. And along the way, we developed a lot of diagnostic platform to detect the pathogen. And the third component is the environment. As you know, that climate change has an effect, and reports have shown there's a lot of heavy metal contamination happening in the water, in the soil, and so on. And this is due to agriculture waste and pesticides. And this actually has effect on global health in terms of toxicity, food safety, contamination, and of course, we look at it holistically from a perspective of food security and biosafety. Now, the three key points, you know that the grants are difficult to be obtained when we talk about biodiversity. So we went on the perspective of trying to understand the impact on how aquaculture and agriculture can impact the biodiversity via genomics. There's a lot of work on deforestation, which had led to threats in conservation efforts. And then the second part is genomics have been used to tackle and give solution for effective management on sustainable use of natural resources. And genomics also can track possible zoonotic transmission within human and animal interactions once the biodiversity is not taken into consideration. So these are the three key points. Now, just to bring you to the fact on human harvest on animals in the wild are always intense, and from whether it's from terrestrial to aquatic habitats, it's all due to economic demand and human dependency. So management plans on harvesting have yet to apply basic principles. In this case, we have used transcriptomic principles with molecular genetic monitoring. The types of genetic changes that we were looking at is alteration of population subdivision, whether the populations have gone through loss of genetic variation, and if there's a selective genetic change. Now, I'm doing a case study on shrimps, and there was this work in 2002, a long time back, published in Molecular Ecology, where they're using mitochondrial DNA. They have actually conducted a population structure of shrimps, and they have identified the different haplotypes in the Indo-Pacific region and right through African regions. So this was the first fundamental base background that we looked at in shrimps. Now, along more than 30 to 40 years, shrimps have been domesticated and it has expanded in terms of aquaculture. But if, as you can see, that the biggest problem in shrimp aquaculture is disease, and it's happening everywhere in across the whole regions across the world, where when they culture shrimps, the biggest problem is disease. So in our lab, these are the questions that we ask. The major challenge faced by the current global shrimp aquaculture production is disease, and the recent outbreak of shrimp bacterial disease, which is called AHPND, has affected the production. But what we propose here is we notice that the different shrimp species, mainly monodon, Macrobrachium rosenbergi, and Latopenis wanemi, exhibit different varying degrees of disease tolerance towards the AHPND infection. So what we propose a hypothesis is, in terms of genetics, there must be something controlling the functioning and behaviors of all life. So the question that we asked here is, what are the genetic factors involved, alterations that lead to variations in disease tolerance? Now, if you look at the research being done across the region, you can see that there's this dogma of molecular biology from DNA right up to the metabolites. And the technology is genomics. Then we look at transcriptomics, then we look at proteomics and metabolomics. And the regions in the genome is, you have the coding region and the non-coding region. And through the regulation, you can notice there's mutations, and then you have epigenetic modification, post-translation modification, and so on. So based on this scope of research, and based on the hypothesis, we notice from phenotypically the shrimps have exhibited different disease tolerance, maybe because of the way it has been domesticated and selected for. So the strategy that we are currently doing is, actually we have three phases in our lab. First phase is looking at establishing shrimp as a biological model, where we study the innate immune system by transcriptomic studies. And the second phase is to establish the role of shrimps to decipher the innate system mechanism to understand its adaptation and survival towards pathogen. And the third phase is to understand how shrimp, being also a natural resources, has its unique antimicrobial peptides to survive and adapt in our natural system. So these are the strategies we do for us to apply genomics in the system we are studying. Now, this is a paper we published in 2019. And we noticed that the shrimps that we use here, it's actually a shrimp that has been crossed between two different regions. One is from the Malaysian genetic line, and it was crossed with Madagascar genetic line. And we noticed this shrimps has ability to clear the WSSV virus, and we noticed that it can shows a quasi-immune response, and it can neutralize the factor, and it can survive. So this is where the breakthrough happened, where we noticed that we can actually enhance using genomics to elucidate the biomarkers related to shrimps. So this is the model we proposed, and we published this in PeerJ, where we noticed that WSSV infection actually triggers the PAMs. PAMs are actually pathogen recognition patterns, and in this case, it is involving, and it has plays a significant role in JAK-STAT signaling pathway, which leads to the tumor necrosis factor. It also activates the serine protein cascades, where it releases antimicrobial peptides, and these are the antimicrobial peptides shown. And in terms of in the apoptosis pathway, it balances and creates an homeostasis balance in using all these innate immune molecules. So this is where we notice that the prawns have innate immune system that can actually clear the infection. So in this case, we just want to show that using genomics, we can actually develop a cost-effective and efficient shrimp health diagnostic kit. In that case, we can further develop probiotics-coated shrimp feed with optimal AHP&D disease inhibitory potential. From the genomics application, we can use it for disease diagnosis, disease treatment, immune priming, and it actually can be used as a baseline for invertebrate immune references. On top of signature adaptation and survival, we can actually study from a selection force comparison, validation across different shrimp species from an evolutionary point of view. And finally, the way we go is to develop a much more effective and holistic approach of treatment and prevention of shrimp disease, which can benefit the shrimp aquaculture industry and, of course, the biological food safety concern that helps can be used to address the global health problems. So this is one of the case studies that we think uh, demonstrate how genomics can be used in the shrimp biological system to address its importance. And this is my lab members, and these are my postgraduate. We come from the lab called Agrigal. Uh, the work we have done, we acknowledge the grant, and of course, we would like to acknowledge the Biodiversity Genomics Consortium for giving me an opportunity to talk. Thank you.